Good morning. Today I'll be talking on beta oxidation. Beta oxidation is a process of breakdown of fatty acid into the acetyl-CoA enzyme. So beta oxidation in the mitochondrial matrix is the major metabolic pathway, catabolic pathway. And beside there are minor oxidation pathway have also been observed. They are alpha oxidation of fatty acid, omega oxidation, and peroxisomal beta oxidation of fatty acids. Fatty acids are oxidized by most of the tissues in the body, but there are few tissues like brain, RBCs, and adrenal medulla cannot utilize fatty acids for energy requirement. So there are following steps of beta oxidation. First is the activation of fatty acid, which happens in the cytosol, then transportation of fatty acids into the mitochondria right from the cytosol, and beta oxidation proper, which happens only in the mitochondrial matrix. So this is the first step of fatty acid oxidation, that is the activation of fatty acid. So it is happening in the cytosol. The fatty acid is now to be converted into the concerned acyl coenzyme A. So it happens via the formation of acyl adenylate as an intermediate, transient intermediate, which immediately converted up into concerned acyl coenzyme A. Only this OH group of the fatty acid being replaced by SCOA group, and it is a very good living group. That's why it makes it a very active. So this conversion is facilitated by the thiokinase enzyme or acyl coenzyme A synthetase, and ATP is converted up into pyrophosphate. Magnesium ions are required, and this is equivalent to, because two high energy bonds are being broken, so it is being taken as consumption of two ATPs. So once this acyl coenzyme is being formed, now right from cytosol it is to be transported into the matrix, because further processing or beta oxidation proper has to take place in the mitochondria, and all concerned enzyme of the pathway are available in the mitochondrial matrix. But there are certain constraint with this molecule. It cannot take it into directly into the mitochondrial matrix. It cannot cross through the barrier of the mitochondrial membranes. So there is a transporter carnitine come into the play. It combines with carnitine, so it form a temporary adduct, that is acyl carnitine. So the enzyme meant to convert this is CPT1 or carnitine palmitoyl transferase 1. Type 1. So, under the influence of this enzyme, the acyl carnitine is being formed. So, in this form, the acyl carnitine can cross through the barrier, and via the translocase, this is one transporter which facilitates the entry of this acyl carnitine right from the cytosol to matrix. So this now once it is inside the mitochondrial matrix, acyl carnitine, under the influence of another enzyme, that is the carnitine palmitoyl transferase type 2, it reverts back or converted back into the acyl coenzyme A and carnitine. And this carnitine is now has to be returned back to the cytosol, because it has done its responsibility, it has done its duties, and it has transported the acyl coenzyme A. Now it will revert back, and it is all ready to convey the another slot of acyl coenzyme A via formation of acyl carnitine. So here now it is in the mitochondrial matrix. Now further beta oxidation proper will happen on this molecule. So CPT1 and CP2, T2, both the enzyme, one is in cytosol, another one is in the mitochondrial matrix, and they're meant to form the acyl carnitine or to break the acyl carnitine. Now, in the third step, that is the beta oxidation proper. So here, from here, this step has been again shown. That is the activation process, conversion of fatty acid into the concerned acyl coenzyme A. So this acyl coenzyme A is there. Now it is entered via the carnitine transporter. It is entered the mitochondrial matrix. So this is the first step. This inside the mitochondria, there are just 1, 2, 3, and 4 steps are there. So we will see one by one. In the first step, this acyl coenzyme A made to undergo a dehydrogenation reaction. So there is an enzyme acyl coenzyme A dehydrogenase, and FAD acts as an acceptor of hydrogens. So in this particular process, the beta carbon loses its hydrogen. So two adjacent hydrogens are lost, and there is a appearance of double bond at the beta position. In the second step, there will be hydration. So this compound is known as a trans-enol coenzyme A. And further, the trans-enol coenzyme A hydratase enzyme. The names are very easy to remember based on the name of either substrate or the product. So it is the trans-enol coenzyme A hydratase. So water is going to be added onto the double bond as per Markovnikov's rule, and the formation of this kind of adduct is there. This is known as 3-hydroxyacyl coenzyme A. Further, another dehydrogenation reaction happens, and 3-hydroxyacyl coenzyme A dehydrogenase is mediating this reaction, and the NAD1 NAD is converted up into the NADH. So this molecule is now further converted up into 3-ketoacyl coenzyme A. So hydroxy group is now converted into the ketonic group. So once this adduct is being formed, finally it is to be broken up via the enzyme thiolase. So breakdown or splicing will take at the from the termini, from the SCOA termini, and the 2 carbon species is being lost. And this is nothing but this is acetyl coenzyme A. And there is a addition of coenzyme A to the moiety which has lost these 2 carbon species. So this is another acyl coenzyme A with the 2 carbon less, and this is the acetyl coenzyme A. So this is one cycle. Suppose we are talking about the splicing or beta oxidation of palmitoyl coenzyme A. So that is a 16 carbon fatty acyl coenzyme A. So for its complete oxidation or for its complete breakdown, there are 7 such cycles are required. 7 such cycles are required for complete breakdown of the palmitoic acid, because 16 carbons, so at every cycle there is a loss of 2 carbon species in the form of acetyl coenzyme A. Right. So, what is the fate of this acetyl coenzyme A? This is a main energy exchanger. Its fate is it has to join the Krebs cycle pathway, and in the Krebs cycle there is a complete oxidation of this molecule, and it converts up into carbon dioxide and water. So this is how there is a complete oxidation of the product of beta oxidation of fatty acid. So if we are talking about the energetics of C16 carbon containing fatty acid, that is a palmitic acid. So how many, it will undergo 7 cycles, repetition of 7 such cycles. Step 1, 2, 3, and 4 are repeated 7 times. So there will be a production of 8 acetyl coenzyme A, and these 8 acetyl coenzyme A will go to the Krebs cycle and completely oxidize themselves, convert up into water and carbon dioxide, and they generate 80 ATPs, because at one cycle there is a production of 10 ATPs. Then 7 Fa, because 7 cycles, so in one cycle I have 1 FADH2, 7 cycle, 7 FADH2s are produced. So 7 FADH2, they are equivalent to 1 point, in the electron transport chain, they are convert up into 1.5 ATP. So there is total production of 10.5 ATP. Similarly, the 7 NADH, which is equivalent to 17.5 ATP from the electron transport chain, and the total ATP production is 108 ATP. And if you remember, in the first step there was an activation and there was consumption, complete loss of ATP in the form of breakdown of 2 high energy bonds. So that is equivalent to loss of 2 ATPs. So we can reduce -2 -2 from ATP from 108 figures. So total net production of a gain of ATP is 106. This is for C16 carbon palmitoic acid fat beta oxidation. If we are talking about the 18 carbon or 20 carbon, similarly in the same pattern we can count the cycles and accordingly we can calculate the bioenergetics. Then regulation of beta oxidation, availability of free fatty acids and the level of free fatty acid is controlled by glucagon and insulin ratio, right? If there is enough free fatty acids, so this glucagon and insulin ratio will regulate the level of this free fatty acids. So glucagon always increases the free fatty acids, and it is all meant because it is lipolytic. So it's all meant to, it will promote or induce the beta oxidation. While the insulin is meant to, it is basically a lipogenic hormone and it is meant to, conserve this free fatty acid into a triglyceride pool. So CAT1 regulates the entry of fatty acids into the mitochondria. You have seen the carnitine acyltransferase type 1, and due to this enzyme only there is an entry of fatty acid via carnitine. So this is at this level also there is a regulation via CAT1. Then malonyl coenzyme A inhibits the CAT1 activity. Malonyl coenzyme A is the product which is formed in the beta fatty acid synthesis. It was the first step in the pathway of fatty acid synthesis in which the acetyl coenzyme A was converted up into the malonyl coenzyme A. So if there is enough malonyl coenzyme A, that will inhibit the CAT1 activity, right? So if there is a fatty acid synthesis, that will inhibit the beta oxidation and vice versa. So now two important clinical conditions related to this disorders of beta oxidation. One is sudden infant death syndrome, that is SIDS. It is a disorder due to the blockage in the beta oxidation. So this SIDS is an unexpected death of healthy infant, usually overnight, and real cause of SIDS is not known. It is estimated that at least 10% of SIDS is due to the deficiency of medium chain acyl coenzyme A dehydrogenase. So deficiency of this enzyme, because baby is only onto the mother's milk and milk contains the medium chain fatty acids. So he is dependent on the beta oxidation of medium chain fatty acid for energy purposes. So if this enzyme medium chain acyl coenzyme A dehydrogenase is absent or deficient, so there won't be, beta oxidation of medium chain fatty acids which are evidently present in the milk. So he is deprived of energy and that may lead to the death. So enzyme defect has frequencies in 1 in 10,000 births. So this is an inborn errors of metabolic disorder of fatty acids. Now then second disorder is Jamaican vomiting sickness. It is actually caused by eating the unripe ackee fruit, which contains an unusual toxic amino acid, that is hypoglycin A. It is some derivative of one amino acid, and this inhibits the enzyme acyl coenzyme A dehydrogenase. So thus beta oxidation of fatty acid is completely blocked, leading to various complications. So it is associated with severe hypoglycemia, vomiting, convulsion, coma, and finally, if other sources of energy are not available, it may lead to death.